hopefully I bought these like.
Okay, everybody, thank you for coming today. If you can come on in and get a seat, we'll try to get it started. It's my pleasure to introduce Dana Nelson. If you guys don't know Dana, he's a forest scientist with the Forest Health Center. Give me the full name, Dana.
Forest Health Research and Education Center.
Forest Health Research and Education Center, which is located partly in this building, so you all may know forestry has some space, lab space, and an office space on the third floor over here, but then also over in the Cooper building. So Dana has brought in a speaker today, and he's going to introduce him, and we'll get underway. Thank you, Dana.
Yep, you're welcome. Thank you. And good to be here. So yeah, I'm with the Forest Service, mostly work out of Cooper. Worked with Tyler Dredden out of plant pathology, and then in the lab on the third floor here. Nurul Faridi is our speaker today. He's also a Forest Service scientist with the Southern Research Station. He's located at Texas A&M University, and he's a cytogeneticist. So he's going to talk to us about plant cytogenetics, bringing us into tree cytogenetics, and what some of the findings he's made, and what some of those applications are with respect to understanding genetics and helping with plant breeding and tree improvement in the forestry side. Nurul has his PhD and master's degree are from Cambridge in England, PhD in '88, focusing on cytogenetics. So he's been a cytogeneticist pretty much from the get-go. He did work with Erie, the International Rice Research Institute, for a year or two, a couple years, and then went with CIMMYT in Mexico. And after leaving CIMMYT in '94, he went to work with the Crop Sciences Department at Texas A&M University. It was during that period of time that some of my colleagues that predated me with the Forest Service research started interacting with him, looking at cytogenetics in trees. And then I believe it's since 2003, then he came on with the U.S. Forest Service, but pretty much stayed put there at College Station, Texas. So with that, we'll turn it over to Nurul. And again, thank you.
I need to get this on.
Yep, just hit the button. Not the phone. Don't hit that button.
Thank you.
Yep,
Thank you, Dana. Well, I work for my life with plant chromosome, and it's still living with plant chromosome. And then there was the first display I wanted to just to give you this overview. That's how the plant chromosome we can make. There's about 30 species. There was the display. So I'm not going to go over that one. Go to the top. Okay. As you know, that's Texas, we are getting a high-speed train. Half of my talk would be like that. And then it's a lot of slides. It's the role of cytogenetics in plant breeding, genetics, and genomics that I'm going to talk about. There are 6 subjects. The alien introduction, that the useful gene from wild relatives, how we can explore and transfer it into a cultivated crop species or tree species. And genome organization, composition, probability determination. You need to know when you start before breeding any line. If you don't know one species that is important in your program, we need to know whether it's a diploid, tetraploid, or triploid. Otherwise, we'll be messing up the whole thing. And we can use this technology to locate the transgene where it is on the chromosome. And then once you know that, then you can strategize your breeding program. And then gene location, markers, and then you can make comparative map, integrated map, And then lately we involved in laser capture, microdissection, and sequencing, and then translation. First is alien introduction. That's, we know most of the land races, it's containing the useful gene, and those genes could be disease, resistant insect, stress tolerance, and quality gene. And here is an example, my graduate research Azilops species. It has a very high molecular weight subunit, and it's glutenate, and that is the most important for bread making purposes. That was the project I did, and my supervisor, he didn't want to give me that one because his previous student failed to get that introduction. As you see, the first lane here, that is the best species that time in 70s, and that is the highest subunit, and you see Azilops ambulata, they have even higher. Now the goal is chromosome 1A of bread wheat doesn't contain that allele, about 50% of bread wheat. It would be good if you could transfer that allele into chromosome 1A. How you could do that in wheat? Wheat is so wonderfully species, and cytogenetically, I think that's the richest species to play with. This slide is showing the bread quality is showing significantly high with when you substitute removing chromosome 1A, putting the chromosome 1U in Chinese pink with background. But the yield is always like this. The alien species has a deleterious effect. So our target is just to transfer this gene from this 1U. This slide shows this is close to the centromere, and we have transferred it here. And my target was to get about 1,000 seeds, otherwise it would be hard to get it. You can use few different systems. That wheat is the 5B system, that pairing regulatory gene that pairs all the A genome, 1 to 7 pair, all the B genome, all the D genome. But when you remove that 5B system, and it's a whole mess, and we have to remove that 5B system to integrate that 1U glue into 1A, and you have to use the 1A, 1U substitution line. That would be the targeted integration. That's what it was. Okay. This slide is the crossing strategy, and I'm not going over this. The total, he asked me to get about 1,000. I have 5,600 recombinant seeds, and each sporate I pollinated 3 times, in the morning, in the evening, following morning, to make sure I have enough seed. So if you see 5,600, probably I pollinated 10,000 seeds, and 3 times it would be 30,000 pollinations. There was a lot of work. I started 3,200, and I got about 60. Out of that, there's 89 glue, and out of 89 glutamine transfer, there was 62 for with 1A, and 27 is from 1U, 1B substitution, and 133 glide. We didn't want that glide, but it happened within this when you use that. But our target was not glide. This slide is showing this transfer of the glutamine subunit into wheat background, and all the recombinant, that 32 recombinant line, I did the homozygous substitution for each recombinant, and then we evaluated that in field trial and then in lab trial too. And we use all the marker. Phylocentric test. Each of the 32 line, I use 1A double dikilo, 1B double dikilo. What is double dikilo does? If the segment is a small insertion, and you would get all this dikilo would pair with the recombinant chromosome. If the segment is large enough, it won't pair. Then you will see one chromosome in the white background. So this slide is showing the 1BL, the single kilo, it pairs with that 1B chromosome. When it comes to that, it was recombinant with the 1A, 1U. Both short arm and long arm pair with that recombinant chromosome, or does it tell? We got a small insertion, interaction, and that's the proof, and rest of the chromosome are ring bivalent. When you get the ring bivalent, that the background is homogenized. And here is showing 1 pentavalent, 4 quadrivalent. My supervisor, he said, look, Nurul, when you sit down with the screening, call me. He came around 9 o'clock. He sat down with the microscope until 5, but 3 break, 2-3 break on lunch break. And it took him 2 days to find out how many chromosomes involved in this translocation. And that 5-bit system is so neat in which you can do a lot of work. Other crop species, stop. So this is showing what the recombinant line we got. And this is the yield trial, and this is the sedimentation test in showing the recombinant 1A, 1U doing really good. Next is pitch location of gene or indicated map. How we do that? We collect the root from the growing seedlings, and the reason is we do not use the germinated seed, because if this plant is genetically important, we can revisit that plant and we can let that plant grow for next generation. That is our strategy to use not the germinating seed, growing seedlings. And we collect actively growing root and do the pre-treatment and then fixing enzyme treatment and chromosome spread. The slide I showed, the first one, different crop species, we do not use the cover slip to spread the chromosome. Just give a thought, those who have some biological and psychological background, we prepare chromosome without discussion. But when the chromosomes are large enough, fine and quick, you need that. But other than that, no. And all are unifocal, nicely distributed. But if you have all these grain and things, if you do not have those, you cannot make anything. You need the instrumentation. That's the key. What is fish is, it's just very simple. You have a probe DNA, you label that, you have a chromosomal DNA, you denature that, both denature and put it on the slide, and it will find the target site and put it on the microscope, and you see where the signal is. If your experiment is chromosome spread is super chromosome spread, you won't get any background, and it would be lighting up like that. So is a fish application, it has a numerous application. You can use it. And the probe we use bacterial artificial chromosome, yeast artificial chromosome. We don't use this in our lab. And you have probe interspersed, tandem, or repetitive clone, retrotransposon, tDNA, or DNA, or LPC DNA, or genomic DNA for fish. This is the sorghum is the bag. You see, one pair of chromosome is on the short arm of the chromosome and on the euchromatin, and it's so discrete, you cannot dispute this. If you see your serological result, and even in the interface is showing 2 dots. And then you build up integrated mapping. The beauty in that integrated mapping, we all know that's all the hardest for short end of the chromosome. But if you show this, there should not be any dispute. So all the marker, this is marker, And from here to here, all in short arm, and from here to here, all on the long arm, nothing in the middle. Look at here, there is 26 marker here. They cannot be oriented from map order. But when you use for cytology, you can orient them and position them. 2 of the 3 go here and 1 comes here. So that's the beauty of cytology. And this 26 probe on packaging chromosome 3 of sorghum. And this is comparative mapping rice and sorghum. It's all towards the end of the chromosome. This Genomics Institute, this chromosome preparation technique, I learned it from the literature, then I played with it. Before making this breakthrough, I was visiting with Perry Gustafson in Missouri. I learned from him, then I applied this technique when I went back to Erie. And one day I used one root and put it on the slide and just smear it, and it did work. Since then, we're using that technique. And here you see all the chromosomes are uniform. And here is critically all 14 chromosomes you can differentiate from the rest of the 28 chromosomes. This is 1B1 translocation. This is 6 addition line. This is leaf cross 24 leaf cross in quick background. Maize trypticum background chromosome, and I was at CIMIT working in next program. My program was to identify the alien addition line in maize background. And when I used that maize DNA as a label and trypticum DNA as a blocking, you see all the 10 light out. And this is one addition line, and that's the event. One of the recombinant line I used here, maize DNA as a label, trypticum as a blocking. When I showed this slide to my colleague, they said it could be an artifact. Yes, it could be an artifact. Then I had a plant growing in the greenhouse, going back and collected the root, did the opposite. Trypticum DNA label, maize DNA block. And you see this one and that one. This is a recombination between maize and trypticum chromosome. There is no doubt about it. Ploid. As I said, we need to know the ploid. And this is aspen, diploid aspen, and it's the diploid eggplant. And there's no cover slip here. And that's tetraploid eggplant, triploid aspen. So it's 3 sets of chromosomes here. And this work I did with Karen Mong, Utah University. Recently, I did this work with a friend of mine. This is the same species, and he thought he did genome analysis, DNA content. This one showed this one 1 picogram 2C, and this is 1.3. And he thought it would be 3 sets of chromosomes. It's not 3, it's 28 chromosomes, and that one 2 chromosomes short. And this is the red color is from the ribosomal RDNA 45S. Is 1 pair of chromosomes, or you can say 1 locus. Here is 2 pairs of chromosomes. The other thing, recently I'm noticing that the ribosomal DNA, it varies from homologous pair. Why is that reason? Why I'm seeing this? And one has more repeat than the other. And I'll be talking about that later. And this species is just located, distributed only in Africa, and it has a medicinal value. If you look at this, is ash chromosome spread with 18S and 5S, and it red color is from the 5S and green is from the 18S. This work with Jennifer Cole, Northern Research Station, because this is for our ash restoration project. The Manchurian ash is resistant to emerald ash borer, that's invasive pest, destroying the ash in an alarming rate. So we got involved to determine the provider because she needs to cause this species to transfer the resistant gene into American ash. What we found, this is 46 chromosome. So American ash, 46 chromosome. The crossing will be an issue. Here is what I'm saying, that with weaker fibers always associated with the stronger 18S, and stronger fibers with weaker 18S. It's across the cell. And why is that? It's probably it's not homogenized or is staying that way. There's one hypothesis could be this preferential selection. Pollen take one, egg takes the other. If not, then it should be homozygous. Why are we seeing the heteromorphism here? Location of tDNA. This is tobacco. 48 chromosome, all are separated nicely. And you see the transgene insertion. This is the pair. And this is not a pair. This is the Depier stain. And what it says is 3 insertion. But molecular analysis showed 2 insertion. So you see the difference. This is not replacing any science, it's adding the information in your research. This is Negotiana, Baldamaniana. We work with Nobel Foundation, and you see the pair of chromosomes showing the insertion. This is Propus chromosome. This is maize, and this is maize. This is heterozygous. This is going to segregate. This one is not going to segregate. Now is what is the time now okay. Our main project in forest service, pine and chestnut. And this slide shows tree from chestnut and tree from pine. They're huge tree. Why the chromosomes are tiny? And why this is like an elephant? Can anybody guess what it has in this system, in this chromosome? It's full of retrovirus, retrotransposon. They're not affecting the species, but they're living happily there. And we will see the genome composition, what these retroviruses, how they're invading and staying in the genome. And chestnut is about 900 and pine is 22,000 gigabases. And you know, chestnut is almost wiped out due to the chestnut blight fungus. And chestnut foundation, they're trying to transfer the blight-resistant gene from Chinese chestnut into American chestnut. That's we got involved in it. Here is 18S and 5S distribution in American chestnut. And this is 2 pair of 18S. One is major, other one is minor, and 1 pair of 5S, this one and that one. Did I pay much attention at this slide? I'll be talking about just only the 18S. If you see, this is we use 4 different lines of Chinese time. We're seeing a different result for 18S and 5S variation. This is a diagrammatic representation. This heterochromatic knob is very bright. Is what I'm trying to say here. It's so compact, and this image can tell that it's highly heterochromatic. And there is a variation of the 18S that I'm coming to the next slide. This is one variety, and that's another variety. This towards the end of the chromosome, the satellited knob, this is heavier, inside is lighter, the interstitial side lighter, and it also shows the Depier stain. Okay. One of the line, it shows the heteromorphism. One has fibers, other one doesn't. And you see the heavy or higher copy of 18S in the other formula. And there is a variation here too of heterochromatic knob and heterochromatic knob here. Look at this one. This is that's the one, and this is this one. And one may say, what is that 5S red cover? When you remove that green cover, and you can see the red 5S signal here, and it tells the copy number variation. Genomic in situ, total genomic in situ, it didn't work that good, but you can see the difference between the heterochromatic, the satellited knob of Chinese chromosome Chinese chestnut, and this is American chestnut, the 18S side. So here, this shows the lighter and the brighter satellited knob. And here is the diagrammatic representation of Chinese, and this is American chestnut. The Chestnut Foundation had an issue about the seed setting. There is a meeting, and they thought in a meeting that could be a reciprocal translation, because they didn't get 12 discrete LG map. It was initially 11, then there's thought it could be a reciprocal translation. How do we prove that? And you get polar sterility, but how much is that polar sterility going to tell us unless we do the cytogenetics? That's what we did with them. This is the packaging spread. The close configuration is the reciprocal translation story. So there is no dispute about this one. And one further studied that linkage, specific linkage group. We put that probe on each chromosome, and we identified that centromeric position, what the centromeric position would be of each of the 12 LGs in chestnut. If you remember that in the sorghum distribution, the integrated mapping and the chestnut integrated mapping is across the chromosome, but sorghum was towards the end of the chromosome. That's the difference between what we are seeing in chestnut versus crop species sorghum. Linkage group A's, about 20 to 25% of the heterochromatic knob and secondary transcription were missing from the physical linkage map. And with this backfish, we identified that it was not mapped before the linkage map. So the reciprocal translation, the map data showed that could be H and L involvement. If it's H and L involvement, when you should expect this when you do the backfish. And we took 7 back and it hybridized in one packaging chromosome, and that's the line drawing. It's showing why this red back is sitting with this green back, the fluorescent. Then the thought could be, it could be a shift translation between these 2 chromosomes. Let's move to fine cytogenetics. This is long ago, then science was not, the chromosome preparation was not there that much. And I'm not criticizing, I'm just showing the difference. This is also enzymatic preparation with cover glass. If you have slides like this, you won't get a good karyotype or ideogram, good measurement. You need a straight good chromosome. It's the same line. This is slash, and this is also slash. And LA11, LA11. You see, this is our spread. You can easily do the measurement. And this is lobularly. And this group tried about 4 years to come up to spread the lobularly chromosome. And this is our lobularly chromosome spread from fine. This is the slash of the same slide, and stem cell is a different filter. And green is a telo signal. Interestingly, why the telo is in the middle of the chromosome centromeric position, and it should be towards the end of the chromosome and also interstitial position. The hypothesis could be is at ancient time, they probably mouse the end chromosome and is forming 24 chromosomes. So we did the karyotype for lobularly fine. We did each and every measurement, 108 reading, and we did a lot of statistical analysis to come up with this ideogram. This ideogram, we're saying that this would be used as a reference karyotype. So the question is, if you have new marker is sitting on chromosome 4 is here, then we'll be saying it is on bean 19 or in bean 19 of chromosome 4. And we have one example. Now this is this is just stereotyping. Okay. We found one unique repetitive DNA that is in bean 32, chromosome 7, and that is from Dan Peterson's work. And it's a repetitive DNA. This is the signal, and this is the homologs. And we did 18S to find out whose chromosome it is. And this is the telo signal, the green signal, and both red and green. And then we showed that the repetitive DNA, it is unique and it's on chromosome 7, bean 42. Genome organization. This probe I got it from creatine law, and you see it's covered all over the chromosome. The interesting thing, the virus, the retrovirus cannot invade the centromeric position. If they invade the centromeric position, then there will be a whole mess of the genome. And also the 18S position, that's highly unique repetitive. And if you see this bluish thing, there is a washing effect, nothing else. The next slide, okay, this one, I was doing the spruce chromosome spread and fish with that clone. I was not getting any signal. I thought I was doing something wrong. Then one day I thought, why not take one root from fine and one root from spruce, put it on the same slide. And you see, I was not doing anything wrong. It's the lobularly fine, they hybridize with that retrotransposon, but not the spruce. Is when the spruce and fine diversion happen, the retrotransposon, it happens after that separation. This is one example, another retrotransposon Penelope element, and this is the first horizontal transfer of retrotransposon from arthropod, from animal to plant. This is an interface, and we work with Dr. Tesla in the PSS department. This is a repetitive clone, and if you see this lobularly fine, and it's one point towards the end of the arm, the gain signal from the 18S as a check. We know that fusiform is a major disease for fine, right, Dana? And is a serious threat in our forest. This is the pine bear, and you see about 50–60% are infested by this fungus. And the project was, we have a test. My boss said you have to identify this locus, who it is on the chromosome. And I said, give me about 3 to 5 kb length because fish has a limit. I got 3 to 5 kb, 3 different probes. I use individual, I use all 3 together. It's all showing blood transfusion. And then this is the deputy image is the signal of this site repeater and interface. So we published this work on that interface. That this is profit. Then I went back and then used other fine spaces. And this is short-lived fine. I showed that long ago, but we didn't realize this is not false positive signal. This is across the cell, the slide. Yesterday I was talking to Dana. He said, tell me about this one. He said, you know what? The short-lived has more multiple copies of that locus, of this effort gene. Well, that makes sense. And it is resistant. Now the question is, are they crossable? They short-lived and long-lived. If they are, then you would be able to easily transfer that resistant gene from short-lived to locally lived. The problem would be almost solved. If we have not done that, we would have not known. So this is LCM, laser capture microdissection. We added this instrument to our program, and this is the brief history. And here, the man is here. I think Jeremiah is here. He has done tremendous work with this LCM. He sequenced individually a single chromosome of salamander. And I think that was the first, to my knowledge, nobody has done that with single chromosome. And that inspired us. If we could do the sequencing with salamander genome, why not if we do it with fine? But the bottleneck is to spread the fine chromosome on membrane slide. You would have ever try it because it looks simple, nothing is simple. Even I tell in seminar, if you go to kitchen to just cook rice, if you do not know how to cook the rice, it won't come out good. So it depends. And then you need the patience and perseverance and keep doing it. And it can be done. Okay. This spread is no cover glass, and this is the membrane slide, and this is non-membrane. And we cannot use the cover glass here. This is nicely separated. And 2 or 3 of my colleagues in our team, they had a doubt about, how do you know you're picking up the chromosome 12? This afternoon, I will be showing some more slides. If you have doubt, just tell me whether you can point out chromosome 12. Then chromosome 12 is the smallest one in the complement and is submetacentric. So anyone can easily identify chromosome 12. There should not be any dispute or authenticity about chromosome 12. Again, you need the instrumentation. This is our LCM. This is our microscope. This is all the PCR work, all done in a laminar flow hood. Someone may say, when you use the laminar flow hood, the laser caps are probably prevent picking up the chromosomes on the membrane slide and collection cap, but it is not. Okay, why fine is a huge genome, and then chromosome 12 can be easily identified. And we made that preparation. And the goal is we do that chromosome 12 amplification, and we have already reference genome. Once we are done with individual chromosome probe, we will be able to do other 11 chromosomes. That chromosome 12 is showing in just with the arrows. I'm not going to go with the technology. We do all the work in the tumor, and we prepare the test slide individually. Each root, so when you do make the suspension, 3 to 1, we use about 5 microliter of the suspension, make the test slide. If the test slide is good, we keep that suspension and put it on the membrane slide. And that's how it is that Wayne Goss guy is very good. He does the work meticulously. I cannot be happier than anybody else. We cannot replace Wayne Goss stuff. He's really good, but he works on his time, his part time. So then we go sit down, make the membrane slide, and then we go do the collection. You see, this is already showing you, see, this is all on membrane slide, just to see which one is the chromosome 12. So those are chromosome 12. Those can, and these two can be, they're sitting together, and they can be easily captured. And this one is showing that one chromosome we can capture, but not the homolog. So this arrow showing those single chromosome can be captured, but the partner one cannot be because there is overlap, and we should not try collecting the other one. So none of this chromosome can be collected from here because it will interrupt with this one chromosome. But if you want to capture the short arm or long arm, yes, you can. Here is all 24 chromosomes in one place. This is a little bit bigger than this one, and it's chromosome 12. And we do the first encircle it, and then isolate it and shoot it, and gets to the cat. I do not know how to play this one. I'm not going to try this. This is an animation. This is a very nice one. And then you can check your collection tube that's for this hole, whether you have the collection or not. So these are the spots on the sticky tag. It says that yes, we are able to collect that and the selected target. And even you can collect the whole cell, and then you check it whether it's collected on the membrane, sticky membrane or not. Then we go do the amplification. After amplification, we run it, we check it. This is the control, and you see we have the span, and it shows the DNA. This is from the primer, and it shows we have able to amplify the DNA. And this is two different source, picoplas and Sigma WGA4 kit, amplification kit. And we found our picoplex does better than Sigma. And we cleaned the DNA with our genome center and did the fragment analysis. And Jeremy is helping us, guiding us to move forward with his knowledge in sequencing. And when you have this distribution, we need to consider those, not everyone. Recently, we have found out even you do not need to stain the slide for LCI. You can use make the laser capture microdissection phase system, avoiding the staining. So we can prepare the slide in a couple of hours. We can go and start collecting the chromosome. So we don't need to wait overnight staining. So I'm giving the summary only this LCM. So we have the protocol for preparing membranes, chromosome membrane slide, again amplification, and we did prove it is specific to chromosome 12. And we are now is in pipeline. We're doing the sequencing. These people, they help to get the microscope. You know, this is a marathon runner. As a Mark Hussey is the vice chancellor, department head, soil and soft. Tom Byram, he was the liaison person from our first service, working with the university because the university put some money for this microscope. And there's the only microscope on campus. And it is in our lab. Here is interesting thing. I think this is the last slide. Read it. And this is pathetic. Where are we heading? Our younger generation, I think, my opinion, it's hard to get money for cytology, but government, in my opinion, should think twice to put some money. Those certain type of research, yes, our younger generation, new generation, they need to learn. The word school means a lot of people, they don't know. You see, out of 12 graduate students, only 2 are correct. And that's sad. This is my summary slide, conclusion. Anybody can make it if you have patience, perseverance, and dedication. Well, that's all. I can take questions.
Questions? Right there.
So we are now in the genomics era. We can sequence everything. You just got a comment that we should put more money maybe to cytogenetics. For me, we can use many modern technologies to sequence the genome, to study the genome structure. We can do lots of things. Why we still need to do cytogenetics?
Good question.
So technology can do the sequencing.
Yeah, so the question had to do with, now that we can sequence the genome in a day or two,
Yes.
why do we still need to do cytogenetics?
There will be some unanswered questions. Those unanswered questions, you need another technology. I'm not talking about cytology. I'm talking about if a technology available there, one must use it. The next question is coming. Yes, we can do the sequencing in a day or 2, yes, but that's coming from all 12 chromosomes as a mass. With LCM, what we're trying to say, when you do the sequencing, would be specific for chromosome 1. And we cannot align that repetitive unit. But this repetitive unit, if that chromosome has, say, 30% or 80% or 90%, we can tell this 90%, this guy is contained this 90%. And then, probably, they find 1.8 million countries is a mess. It's just another tool. If we have some issue, this tool, if that tool can help our research, why not use it? Because as I showed the last slide, the chromosome, from the DNA coming, is coming from the chromosome. If I do not know the chromosome, am I ignoring my mother? No, I'm not. I'm sorry, it's getting a little bit off the line. I'm talking about myself. It's the connection, nothing else. That's my point. Yeah.
So how can you adapt this kind of study to the genomic sequence we have? You have to do some, for me, connection between this and the for this alone, it's so low throughput.
Oh yes, it is low, and everybody cannot do it. And I'm not saying that there's a few things. I'm not saying we adopt everything. in medical science, in cancer research, this technology is playing a significant role. And they're using it. When you have a infected cell, you want to get that infected cell isolate from that tissue and then analyze it. But I do not know where genomics thing can isolate that individual cell from that area.
Question.
So the retroviral accumulation, retroviral DNA accumulation of loblolly pi, you mentioned. So I have a couple of questions about that. So one, are those many of the retroviruses still existing in nature? First question. Is that no? Or are they ancient retroviruses that are long extinct?
Well, that the Penelope one, when the pine diverted from the other groups, that's retrovirus. It's the beetle. It's the, I think it's the insect arthropod. It came from the arthropod. Arthropod was in the tree, and it found in arthropod, and that's invaded from there. And its retrovirus is in the environment, and the question is, why is it they got in pine? I don't know.
So it's much of the genome, right?
Yes.
So is it mostly one or a few retroviral sequences or genomes, or is it many different kinds of retroviruses that are found in there?
Well, they're saying Zipchi-like, and there's one group that the one we published who said RT.
Yeah.
Yeah.
Copia.
Yes.
Yeah, there's several
Several.
kinds or different classes that they haven't all been delineated.
They're all from arthropods?
No. We only know the Penelope one from arthropod. Other ones do not.
Plants don't have retroviruses, right? So the retroviruses are all animals, is that right?
No.
Yeah, I'm not sure.
So the retroviruses normally go in and out of genome, right?
Yes, but in pine they allow it. They're staying there. It's, I don't know. If you see the tree, the chestnut and pine, I don't think so they're affecting that tree that much. They're happily living there. They found that way. When they sit there, they can be multiplying over and over. And the interesting thing is the retrovirus in pine all over. Where is that gene acting? I have a nice packaging slide. I didn't include it here. It shows the one chromosome. It shows the heavy and the dark. The other one is lighter. Why is that? We should expect more or less same. Many places, and some places I can see the loop. The chromosomes are not in.
We talk about retrotransposon or retrovirus.
I didn't get it.
Retrotransposon or
Virus.
virus.
That's a different thing.
It's two different things.
Yeah. Well, I think Penelope was a retrovirus, but I think the other one's a retrotransposon.
Yeah.
And the other one. Fine.
So how far along are we in getting blight-resistant chestnuts?
Yeah.
They're coming along, and are they still staying with the 3-gene system?
Yeah. So there's they're running a back cross breeding program, and those some of the images the rules show on it is working with that. introgressing resistance from a few different lines of the Castania mollissima, the Chinese chestnut. And so right now, within the Chestnut Foundation's breeding program, they're evaluating the third generation backcross parent trees for how well they captured the basically 3 different genomic regions that are responsible for blight resistance. They're kind of sorting through those to try to figure out which of those lines in that third backcross generation have the resistance. So that that's ongoing, and some of those lines look pretty promising. And then there's also a genetic engineering component that has been led by researchers at State University of New York in Syracuse, SUNY in Syracuse, and they have an OXO gene homolog ortholog from wheat that certain event in American chestnut provides really strong resistance to blight. So that's being evaluated through under permit with APHIS and FDA and EPA for potential release as well. So that's, pure American chestnut is engineered with a gene that's providing really strong resistance to blight.
The third backcross would still be 1/8 non-American chestnut, right?
Yeah, it's third backcross and then the intercross selection. Intercross. So it's about 15, 16 American, 116 Chinese, about where it comes down. And so there's a pretty big effort now with integrating those 2 programs basically, where the transgenic event is in a single genotype of American chestnut that was used through all that research. And so now they're looking to cross that into multiple genotypes of American chestnut that increase the genetic diversity that transgene is located in for producing a diverse set of trees that can be used in cow planting for the restoration effort.
Think they'll allow the transgenic ones
out
in nature?
There's some, they're fairly optimistic. They've done a lot of testing, produced a lot of data, that's been requested by the different agencies from a state biosafety standpoint. Everything looks good, but it comes down to being able to persist through the regulation process against counter suits by environmental groups. There's still, obviously, there's still pushback in that respect. So it's a matter of just trying to continue to work that process. Don't know, but they're maintaining their optimism. Jeremiah.
This result of this difference between the longleaf and shortleaf pines in terms of, I assume it's a copy number difference between the 2 that confers the pine resistance, or that stops to confer the pine resistance. It makes me wonder, if I were to be looking for a human that had a duplicate, had a copy number variation, or if I hypothesized that some copy number variation was contributing to resistance in a human, I would just go look at a lot of people and find the one person who has, or find the people who have a duplication or that have a modification of that locus. So it makes me wonder if this justifies sort of trying to screen longleaf pines for like the lucky pine that maybe has a duplication of this locus, or that sort of might have an extra copy of this gene.
Yeah, that's interesting. So it's loblolly pine and shortleaf pine.
Okay. Sometimes breeding things, plants are different than animals, right? You can do these distant processes.
The locus was mapped in loblolly pine because we found some trees that that segregated for resistance. And so we did classical linkage mapping. And then as Darul explains, some of the markers were elongated to use in fish, just to take a look and see what it looked like in the fish. And my understanding was that when you did the fish with the FR1 related probes, I may have missed this because my understanding is a little different. You didn't get any signal in loblolly, or you got signal everywhere?
Signal everywhere. Loblolly and
single probe or the cocktail of three?
Single and cocktail too.
Okay. Yeah. And so basically, in shortly, fine, what you're saying is that locus is all contained in one locus and it's not spread out throughout the genome. Yeah. So I misunderstood that clearly there's a very specific difference between those two species in the distribution of that locus, and that locus we know in loblolly pine confers a very distinct resistance. And it's a gene for gene resistance. So there's lines of the fungus that will overcome that resistance. You know, heavy virulence, staph as well. But shortleaf pine tends to be extremely resistant to fusiform rust, and loblolly is variable from anywhere from relatively resistant to quite susceptible to phenotype. There's quite a bit of variation. So yeah, that's an interesting result there, but you have to be kind of tracked down on what's going on between those species.
And the fusiform rust, is it a recent disease or is it sort of generally persistent?
Native disease.
Okay. So maybe there's some downside to being resistant.
Yeah. It became extremely problematic through the early establishment of the southern pine forestry in the south. Plantations were established really because of rust, well, like oftentimes do. Yep. All right. We're up against the clock, so thanks again, everybody, for coming. Yep.